So today we're going to talk about the scientific method and experimental design. The scientific method is used to find answers to questions or to solve problems. You will find an outline for the scientific method notes and experimental design notes on Canvas, so please make sure you have that ready so you can record your notes. You could also record notes in your workbook. So first we need to identify our problem. What do we want to know? What are we trying to figure out? Sometimes these are written in question form. Next, we need to research the problem. As we research the problem, we're trying to figure out what we already know, what has been learned before. There's lots of resources for this: books, journals, periodicals, and of course the internet. We do want to make sure that we cite all sources and keep track of where we find our information. It's important that we don't plagiarize and use someone else's work without giving them credit. Once we've conducted our research, we're going to form a hypothesis. The hypothesis is our predicted outcome, what we think is going to happen in this experiment. It doesn't have to be accurate, but it should be our prediction, how we think this experiment will play out, what we think will happen in our experiment. Sometimes the hypothesis is written as an if-then statement, but not always. Keep in mind that sometimes your experiment will prove your hypothesis, and sometimes it will prove it incorrect. Either case is fine. The hypothesis allows us to have a direction and a focus for our experiment. Once we have our hypothesis, we can now actually run our experiment, and we have a procedure for our experiment. This is the way that we're going to test our hypothesis. We need to design the experiment to test that hypothesis. We do want to make sure we record the steps. They should be organized and numbered or bulleted so it's clear what you performed, how you went about taking those steps, and someone else can then repeat that, or you could repeat that again later. Your materials are also listed in a bulleted list. As we run the experiment, or as we wait for the experiment to finish, we're going to record our data or our results. The data is going to be gathered for what happened in that experiment. What occurred? What kind of data can we collect? Data comes in two forms: quantitative, which is number-based, or qualitative. Qualitative is descriptions. Both quantitative and qualitative data provides very important and necessary information. In our brine shrimp lab, we used quantitative data because we counted the brine shrimp, but we could have descriptions, maybe color changing or photographs or drawings, and that would be qualitative data. Both are very useful. When we do collect our data, we're going to organize that as best we can. So for example, we might make a chart or a graph or a data table. We might also include images, illustrations, pictures, drawings. From our data, we will then form a conclusion. So our conclusion is an analysis of our data. We analyze the data and then summarize what we have found from our results. For example, in our brine shrimp lab, maybe we would say which of these salt concentrations was the best environment. Here we'll link back to our hypothesis. Was our hypothesis correct or incorrect? Not because we want to be right or wrong, but because we want to find out some information. We can always adjust our hypothesis and rerun the experiment with that adjustment. We do want to make sure at this point we look at all possible sources of error. Some of those are human error and some of those are not. What can we do differently? How can we improve this experiment to run the experiment again and hopefully next time a little bit better?